and welcome to my poster presentation. My name is Szilárd Sajgó, and I will show you how your organoid research can greatly benefit from high-density electrode recordings. Why would one want to study human organoids, and what is their relevance? Well, one of the major challenges of neuroscience research when trying to develop new therapies, or even during performing basic research, is the reduced translatability of animal models and two-dimensional cultures. And how can organoids really improve on this? How can they help? Well, their three-dimensional architecture is critical to organ physiology and functions, allows for the development of cell types, thus it recapitulates aspects of in vivo physiology more accurately than its two-dimensional counterparts or even sometimes animal models. Now, what aspects of biology can one study using organoids in combination with microelectrode arrays? There are examples in literature where organoids and MEAs were used for drug discovery, for drug screenings, or they were used to study disease mechanisms, to screen toxicity effects of different compounds, to study regenerative medicine, to study different aspects of development in these organoids, or even to study infectious disease effects on these organoids. Using MEA technology, one can record from the organoids in general two outputs. One is the LFP, the local field potential, this being the electrical potential recorded in the extracellular space of your organoid, or you can record, of course, the extracellular action potential of your neurons in your organoids. Here at MaxWell Biosystems, we provide high-resolution single and multi-eval microelectrode array systems with 26,000 electrodes, enabling the users to record, stimulate, and analyze organoids at really single-cell resolutions. Our systems also come with predefined assays that allows you to detect the overall, the network, and the single-cell electrical activity within your organoid tissue. And as you can see in this small video, you can really retrace the entire electrical activity of a neuron, of a given neuron. As I mentioned before, we have assays, and with the activity scan, you can record the overall activity of your organoids and analyze its basic response properties. You can analyze the firing rate, their amplitude, and so on. And unlike in conventional low-resolution MEAs, these systems allows you to record your organoids' electrical activity independent of placement, because there is always an electrode at the correct position, and the really low noise readouts facilitate the extraction of neuronal activity features at a very precise rate. Now with the network assay, you can access the network bursting activity within your organoids during normal or during drug application. And for example, as you can see on the left, you can see the baseline network activity of a cerebral organoid, and you can also see that by applying CNQX and MK801, you can, for instance, determine that both glutamanergic and GABAergic neurotransmission contributes to the synchronized network activity within your organoids in question. And thus you can really study different processes like the GABA switch. Now, the network assay also allows you to characterize not only single organoids, but also fused organoids, so-called assembloids. And as evident by the rasters on the right, you can really see that the network activity greatly depends on the type of assembloid that you're recording, whether you're recording single cerebral organoids, or you're recording ventral and forebrain fused organoids, or ventral forebrain or cerebellum fused organoids. And as mentioned previously, the high-density electrode array allows for detailed extracellular mapping, and it allows you to easily extract the footprints of single neurons by a process called spike sorting. In conclusion, the MEA systems from MaxWell Biosystems will allow its users to monitor labor-free and long-term neuronal activity of your organoids. It will allow you to collect novel and reproducible readouts, data. It will allow you to compare functional phenotypes between organoids. It will allow you to measure really drug effects on your organoids, and it will allow you to study neuronal circuits at really a single, even subcellular resolution. And with that, I would like to thank you for your attention. And if any of this has sparked your interest, it would be great if you can schedule a call or even potentially a demo with us. And for this, you can use our Calendly link as mentioned here on the slide. And with that, I would like to thank you for your attention.